So I have mentioned colocalization analysis briefly, but what is colocalization? It's maybe I must just put a disclaimer here. It's one of those interesting tools in your toolbox, which I think there was initial excitement about it when it was just first introduced and people were using it, and then there was some misuse of it, and then it led to some poor results or poor interpretation, and then it fell a little bit out of favor, and then some people felt that some of the metrics are a little bit inaccurate or not very descriptive, and I've tried to, through some of my own research, try to improve some of this interpretation of these metrics, but it is one of those methods which you definitely have a risk to read more into it than is really going on, but it is also giving you a lot of insight into what is actually going on with interactions within the cell. It's a very holistic view of interaction, is the description of it, or estimation. So what do we have? We've got all of these different molecular structures within our cells. They are usually tagged individually with different fluorescent probes, and then these fluorescent probes hopefully bind only to the structure of interest. So if we see these fluorescent probes occupying the same space spatially, like they're in the same XYZ coordinate space, then it should indicate that these two molecular structures that are sitting behind those markers are interacting with each other in some interesting or sensible way. So the point that we're trying to do with colocalization are two different distinct molecular structures interacting with each other, or at least occupying the same space, which means that there might be the one facilitates something related to the other one. Now there are two types of things how this can be considered. It could be that two structures are just happily moving past each other, but there's actually no interaction. You can call that co-occurrence, that the overlap in the intensities of these probes should most likely not be proportional, so the one might be bright, the other one might be dim, because they're not actually binding to the same location, they're just moving past each other, for example. Another one is correlation, where if they are really bound together in some interactive way, then you would expect the intensities of these probes to have similar variation for the same location, so that's correlation. And colocalization can be analyzed in various ways. The most common way is quantitatively, where you calculate some colocalization metrics, and we're going to have a nice quick deep dive into these things. Another way, which definitely should be part of the consideration, is you analyze it qualitatively, where you look at the sample visually. Now, visually, as I was just saying, can lead to a lot of misinterpretation. I developed a new tool that maybe gets rid of some of that misinterpretation, but we'll get into that a little bit later. So, where did the misinterpretation problem come from? Well, first of all, from not very specific probes. So if you've got two probes, in this case, I'm not going to comment on the biology of this, but the green and the red one, and you want to know, are they interacting? You basically look at the two images and it's like, okay, well, green plus red is equal to yellow. That's how light works. So therefore, wherever I see yellow, there's interaction. So then there's obviously interaction basically everywhere in these samples. So these then are strongly associated to each other and everything. But there's several problems with this. First of all, you're estimating the degree of interaction based on your visual perception of how yellow something is. Now, visual perception, I'll make that point stronger just now, is not uniform for all different colors. For example, if you see blue, you think it's dimmer than it really is, because your visual perception isn't very strongly associated with blue. Red and yellow, these become more, they are perceived more vibrantly. So we think they're brighter than they maybe necessarily are. And remember, there is no such thing as an image. It's only a set of values sitting behind all of this. And these values are actually the thing that we should consider, not our visual perception of a color, because there are two probes that are interacting. This is not an entirely invalid approach. It's still good to, like an initial gut feel, am I seeing some yellow? Okay, there's some interaction. It's just, it can't be the final conclusion. Now, to drive the point a little bit home about our visual perception of things, can anybody tell me what the difference between these three images are? Like, what is it the same image? Is it different images? Were the different probes used? Or just what do you think happened here?
Looks like some of the structures of different probes.
Different probes, okay. Any other thoughts?
It looks like the same thing with a different filter.
Or just different allocation of color.
That's exactly what it is. It's different allocation of color. A better way of saying it is I change the hue of the image. So if I take this image, I've got it now in some other software, where I can easily change the hue. So you remember hue, saturation, lightness, or brightness. We discussed this yesterday. Hue is just basically this circular thing where I can just continuously change between the different colors, and that means that I'm assigning different colors to every color. I'm shifting this entire image around on this circle, basically. So as I shift this around, you'll see that, hey, look, there are structures there. I couldn't see them before, but wow, they're actually there. And then if I go to here, it's like, oh no, there are actually no structures there. Nothing has changed to the underlying values. It's just the visual perception that has changed here. So visually looking at colors can lead you down very poor interpretations if that's all that you're relying on. And it's just good to be aware that our visual perception is not uniform in that way. This was actually highlighted, or this problem was highlighted in this paper from 2016, where they kind of made this point that the probes that we usually use for microscopes give out quite saturated colors. So it's quite red or green or blue. It's very high saturated colors. But our visual perception of these high saturated colors are not uniform. This is the luminosity, or how an estimation of how bright we perceive them. So you can now confirm this for me, but we visually perceive blue as much darker than we would perceive green, for example. So this looks, if I just look at that, it seems brighter than that, but the actual intensity value behind it is exactly the same. Where if you were to make this uniform according to how we perceive brightness, then you would say, oh, all of these colors look vaguely the same brightness. Maybe there's still some variation, but vaguely the same brightness. But that's not what we get typically from our microscopes. We don't get all of these washed out, muted colors. We get relatively bright, vivid colors. So this is a problem if we rely on perceived luminosity to decide, make some conclusion. All right, so visualizing colocalization then. If we don't want to look at color, what can we look at? Now, I will make a point about thresholding a little bit later, but when we capture data, there is always some, I don't know what the right word is, but bleed through, I don't know if that's the right word is, but if I'm capturing green, there's going to be some redness in that green, and that is not actually red signal, it's just due to the way the sensors work, it's going to pick up some red. So I want to get rid of that so that I actually only look at the green signal overlapping with the actual red signal. So I need to set up some thresholds, and we'll look at that a little bit later, on the green channel and on the red channel, where I say everything below this is not even being considered for colocalization. Both intensities have to be above a certain threshold before I would even consider this as colocalization. So if you've now set up these thresholds, which could be done a little bit manually, iteratively through your own perception, or there are automatic ways to calculate this, or there are image-based ways to determine what is a good threshold. I'm not going to discuss all of that, but this could be, for example, the result. You can either take your image and then just say wherever a voxel, this is not three-dimensional, so it's a voxel, wherever it's considered colocalized, I'm just kind of color it in white. So I'm not relying on my visual perception of yellowness here. I'm saying if it's actually colocalized, show it to me in white, and then I can see where the dense regions are. So there are more colocalization happening in the perinuclear region than in the membrane region, for example. So it gives you a bit of a gut feel for that. Another way is to say, okay, let's extract all of those intensities and then show them actually in the color. Now here again, you need to be careful about the perception of color, but you can kind of still see a bit more of a feeling of density, and you can see the shape of the structures and everything. So that's another way. You can also just do the same thing, extract it, but just show it in white if you want to get rid of that perception. I think all of these are lacking, so I developed a new way to visualize this, which I'll discuss a bit later, but for now, this is a common way to visualize it. So in order to be able to discuss colocalization, it's important to understand how signals relate to each other. So if I take every single voxel in my entire image and say what is its green intensity, for example, and for that same voxel, what is its red intensity, I can then plot that onto a scatter plot. So it's a green, red, there was a voxel that had 150 intensity, so there's one dot on my scatter plot. And I do this for all of my voxels, and then eventually there are certain combined intensities which are more frequent than others, and I would then highlight them a little bit. So I would, for example, in this case, if there are more voxels, then for that combined intensity, I would show it as yellowish color, where if there are fewer of them, I would show it as blue, for example. So a scatter plot visualization gives you a good understanding of the spread of your intensities between the two channels. And then if I look at the spread, I can make some just general conclusions. So if they are strongly correlated, in other words, if the one intensity goes up, the other intensity also goes up. If the one goes down, the other one goes down. That's positive correlation. So that means that there must clearly be very strong interaction between these two because they are varying in their intensity together. If they are entirely unrelated, they are basically just all coincidentally overlapping, there's not much structural similarity, then you would expect to see something like this, where it's just there might be some general overlap between certain intensities from the one to the other, but it's not very structured. But they're basically independent. If you see something like this, it means that the two channels are basically independent. If you see something like this, this could take many forms, but where it's very spread out, that would typically be zero correlation. In other words, knowing something about the one tells you literally nothing about the other one. If the one is high, is the other one high or low? It's like, I have no idea. There's too much of a spread there. Okay, and then you get negative correlation, which I've personally never seen in imaging in biology, but theoretically, negative correlation would be that if the one goes up, the other one goes down. So yeah, here are some example images with their related scatter plots. You can see if the two structures are basically perfectly overlapping, you would expect a very strong positive correlation. If structures are somewhat structured, well, these are actually two independent ones, so there's not. I don't really see much overlap between the two. Some yellow streaks on some of it, but not really. So it's somewhat random, mostly unrelated. And then these are two structures which are so independent that the one has nothing related to the other one. If the one goes up, I don't know if the other one is going to go up or down. So this would be then uncorrelated or zero correlation. Okay, so you just take a moment to take this in, overlap, how it relates to a scatter plot. Right. I've already shown this, but this is just another way that you can consider visualizing it. You can assign to the scatter plot the color of the probe that you used, and then you can say, well, I have more of an intuition of where the overlap is. It's in this lower region, or you can just show it as a heat map. You can kind of see already here where the thresholds are applied. You typically want to cut out this. If you see something like this, that means that this overlap is due to the fact that the probe was not maybe pure green, it was capturing some in the red channel, and vice versa. So if you see something like this under the microscope, after you plot the scatter plot, usually this would be a good indication, is I should put a threshold there to cut this out, because all of this is actually not true colocalization. It is the way that my sensor captured it. That's what you can kind of see what I did here, is I cut out this bottom portion before I showed the scatter plot.
And that threshold you will also determine visually.
Yeah, so one way you can do it is to determine it visually. I'm going to more discuss a way to do it automatically, but yeah, you can definitely do it visually. What I kind of prefer is I would look at an image like this. I would have its three-dimensional representation in front of me. So I actually wrote the software myself to show it to me three-dimensionally. And then I would play around with the thresholds until I look at the scatter plot and say, okay, the threshold seems to be in a good place visually. Does it seem like it's cut out all of the random noise, which is not actually overlap? And then I would say, okay, cool, this looks like a good place for the thresholds to be. Select that. But like I said, automatic ways are fine. Often the more time you spend into it, the more accurate you end up getting your statistical significance. Yeah?
The other thing that we do is based on the company, but we have single stains. I usually require people to have single stains, especially for colocalization. And I image it as single red, and you can see there is no green in there. So then you can basically see the line where you will still have that spread. You see there's the pixels that's kind of like there. It's still a bit of a subjective where exactly do I put it on. If you know there's no red, it's a
good guideline.
Pretty close to what the difference is between them.
Yeah.
And you'll do it for the other color as well. So you'll get the line for this, you'll get the line for that, you'll get the line for
Yeah, and maybe just.
one way, that's one.
Yes, but maybe just to remind you why this happens, why you get this spread, even, like let's say for example the red is not here, you would still get the spread of the green to be somewhat having some red captured. It's because the sensor, the red, green and blue elements, they have some quantum efficiency, so they activate over a wide range of intensities, and the red sensor activates a little bit when there are green intensities, which is not what you want, but it happens. It's how the sensor is built. So then, of course, if you capture something that's perfectly green, the red sensor activates a little bit because of its quantum efficiency, and therefore you get this type of a spread. And you want to cut that out because that's not actual data. sensor error, if I could call it like that. All right. I'm going to show you in Fiji how you can do colocalization analysis. I'm going to first show an example, and then, yeah, unfortunately, I don't think we're going to have time to do it together, but I'll do it very quickly. I gave you the script. There's a macro that I put into the folder. You can run the macro, and it will do everything magically for you without having to put any time into it. This example in the slides is based on certain cells which I no longer have access to, or I never had access to them for that matter. This is someone else's slides. So I will demonstrate this on HeLa cells, which you can use in the sample, which is in the sample folder. But yeah, so there's this plugin called Coloc2, or Colocalization 2. When you open it up, it shows this, or it asks for you which image is channel 1, which image is channel 2. These are the two images you're obviously trying to calculate the colocalization on. Then there are lots of parameters that you can choose. We'll discuss most of them as we go on, so you don't need to worry too much about this, or not understanding it right now. And then you just click OK, and then it will automatically, in this case, it uses this costas. If you, where is it now? There, threshold regression. If you select costas, then it will automatically determine where the optimal threshold is for the two channels. And then it might generate something like this. It's like, okay, well, after thresholding, you get this, so it's already removed all of the low intensities. It's basically like a segmentation. It's an automatic segmentation that it does first, or masking, it's a better word to use here. It first masks the data so that the background is removed, you only have the signal left, and then after that, it does the colocalization analysis. And then you can see, for example, here's the scatter plot. These two structures are clearly mostly strongly positively correlated, and then you get all of these metrics that's also calculated. So first of all, what are these metrics and why are they important? I'll discuss that now. But the scatter plot feature, and this is what I think it was sent out in the email beforehand. If you want to see the scatter plot, everything else works, but the scatter plot is broken in the newer version of Fiji. It no longer shows you the scatter plot. It just shows a blank space. So if you actually want to see the scatter plot, then you need to download an older version of Fiji, like early 2022 still works, late 2022 no longer works. So somewhere someone broke something. They're aware that it's broken. Nobody's fixing it. I don't know. Okay, so I'm first going to discuss all of these metrics, and then I'll do on that HeLa cells example. So as we've been saying, the color itself is not very helpful to interpret what's actually going on there. So we don't want to rely on subjective color interpretation. We want to rely on something objective, metrically comparable between different sample runs, between different treatment groups. We want to have some value we can assign to colocalization. How much interaction is there? And then see if it statistically changes from one treatment group to another. Okay, so the most common metrics are these four. You get Pearson's correlation coefficient, and then Manders overlap coefficient, and then two Manders correlation coefficient formulas. I'll go through them just now, but just absorb them in all of their glory. Pearson's correlation coefficient. What this thing is trying to calculate is how correlated are the two signals when you consider it around the mean. I'm going to now with my hands try to explain this. So if you have your axes, maybe I can do it on the scatter plot. That might be better. If you have your axes X and Y, channel 1, channel 2, then for each of these channels, if we were just to consider one of them individually, there would be a certain mean value. So I'm just going to now thumbsuck this a little bit. Let's say that for channel 2, the mean sits somewhere there. And then for channel 1, the mean sits somewhere there. So the joint mean for the channel 1, channel 2, I don't know, let's put it somewhere there, where that little dot is. So then what Pearson's correlation coefficient says is if channel 1 goes higher than the mean, does channel 2 also go higher than the mean? If channel 1 goes lower than the mean, does channel 2 also go lower than the mean? It's trying to find this general relationship. And if they go in the same direction, you have a positive contribution to Pearson's correlation coefficient. If the one goes up and the other one goes down, which means it goes in these opposite directions, then you have a negative contribution to Pearson's correlation coefficient. And also, if it is far away from the mean, it contributes more heavily than if it's close to the mean. So you want to know on the variation of extremes, how much does that actually affect the similarity? So it weights those slightly differently. And then you get this formula. So this R stripe is for your red channel. The mean G stripe is the mean for the green channel, and then every individual pixel or voxel, what is the red intensity for that voxel, what is the green intensity, and then you just plug it into this formula and you get the final answer. The way this formula is set up, it ends up that it primarily only considers the intersection between structures. If there's no overlap between any probes, I'll show an example just now. If you've got green entirely by itself and red entirely by itself, Pearson's correlation coefficient will estimate close to 0. It won't be exactly 0, but it will be very close to 0, which means that there's no reasonable correlation between the two. Okay, I did kind of say this, but if they are perfectly synchronized, the value ends up being exactly equal to 1. That means that there's perfect correlation, everything is aligned. Anything between 0 and 1 is positive correlation, which means that they are heading in that same direction, but they are maybe slightly, there's a bit of a spread around it. And then if it's the negative correlation, which is that one that I've never seen, then the value becomes -1. So this is somewhat of the consequence of that. If you've got two structures, they are not overlapping at all, Pearson's correlation coefficient will get close to 0. I believe, I did this quickly now just beforehand in my head, I believe the answer will actually be, the correlation coefficient will be 1 over n, where n is the number of samples that you have, the number of pixels, voxels in your image. So if you've got a 1,000 by 1,000 pixel image, it's 1,000,000 pixels that you have, so 1 over 1,000,000, which is essentially 0. So yeah, it will be close to 0. For interaction, that is now where it starts to come into the picture. There it does actually produce some values, and it will be non-zero. Okay, Manders overlap coefficient. There are two types of Manders things, overlap coefficient and then the correlation coefficient. Here you see that it's basically the same formula, except I don't consider the mean. I just consider the ratio of intensities, basically. So it's how can I say this now? It's all of the intensities considered together. It's not just where they overlap. It's the one can be 0 in one place and have a value in another place, and then it will just make that term 0. But it's going to consider all of the nuances of the entire space, not just where it intersects. And you can see it also here for the specific channel ones. So this is now combined one. This is now looking at a single channel. So if I look at the red channel, for example, it's basically the percentage, the weighted percentage of voxels that are overlapping. I think that's a helpful way of thinking about it. So if I've got my entire image, just first of all, what is the percentage of overlap? How many of the red voxels are also overlapping with green voxels? And how many of them are not overlapping? If they are not overlapping, it contributes to 0. If they are overlapping, then I just add that to my formula. Now, obviously, this is an intensity value, so it's weighted by intensity. So it's not just percentage of overlap. It is weighted by intensity percentage of overlap for the red channel and then for the green channel. Now what's helpful here is it might be that your green channel fills the entire image, and then red is very small, and then you would say for the green the amount of overlap percentage is very low because there are only small little green, red dots everywhere. But for the red channel it's very high because all of the red dots overlap with green, so it gives you a bit of a more nuanced perspective per channel what is actually going on here. I think these types of examples get to the point a little bit better. If I have a situation, red and green, perfect intensity, they're overlapping. You can see basically 50% is the way you can think about it. 50% overlap for all of these. If I was to change the relative intensity of one of them, the percentage overlap doesn't change because it's, when I say it's a weighted intensity, it's still weighted by the channel itself. So it's the ratio of the intensity divided by the actual intensity of that channel. So if I make it dimmer or brighter, Manders is not affected by that. And then for the Pearson's correlation coefficient, you can see if there is just a little bit of noise in the one, in other words, the one is now going up and the other one is not going up by quite as much. So they're not exactly aligned. They're somewhat aligned, but not perfectly. Then Pearson's correlation coefficient says, okay, it's a lower value, and then the more noisy it becomes, in other words, the more the intensities don't become aligned, not correlated, then Pearson's will keep going down, while the percentage of overlap is still the same. It still stays about 50% overlap. And then similarly, if I was to then add more and more green area, then the percentage of overlap goes down. You can see for the M2, that green percentage goes down. Also the global one, which takes both into consideration, also goes down. But for Pearson's, it stays the same because it only really cares about the intersection. It doesn't care about how much of there is. It's only really looking or weighting properly where they are overlapping. So this is hopefully a bit of intuition behind it. But we've been talking about this threshold quite a lot. How do I find the appropriate threshold? And then the second question is, even if I have the appropriate threshold, how do I know that the overlap that I'm getting some metric out? I'm getting a value of 0.45. is the 0.45 just coincidental overlap? So coincidentally there was some intensities that overlap, or is this actually structured overlap, that the structures are matching with the other thing? These are two important questions that we need to address. So the first one is, how do we find an automatic threshold? Now there's this Costas approach, which is pretty standard nowadays in this type of analysis, and how it works very quickly is you start by assigning your thresholds basically to the maximum value. You say, I'm throwing away all of my data. Channel 1, let's say the green and the red thresholds are at their maximum. Okay, then I take everything that's below that, and then I say I throw the top part away, and then I say everything that remains at the bottom, what is the correlation for that? Now, initially, if there is correlation, you will see that there will be, let's say, a correlation value of 0.4. Then you shift this threshold down, you throw everything away above this line now, that's gone. Then you say, what is the threshold, what is the correlation now? Well, this part was nicely correlated, and that's now gone, so now it's a little bit less correlated. Okay, and then you keep shifting the threshold down iteratively until you get to that point there, and then you say, okay, well, now basically I'm only looking at all of these uncorrelated regions. All of the correlated parts are now thrown away, and then as soon as you get to a correlation value of 0, which means that there's no structured correlation anymore, then you say, okay, this is a good place to choose my thresholds. So this is an iterative approach where it progressively moves this thing around until it finds what is a good choice for red and green. It's by no means perfect, it's an automated one, but so it is technically better to still do it by hand, choose the thresholds. But if you just need to have a quick analysis, a feel for your data, then this is a pretty standard way of doing it. Okay, now I'm going to go through this a little bit quickly, but if I've got these two channels, there's my red channel, my green channel, and if I extract only the parts that are actually in overlap, I would extract these yellow regions. Those are my correlated, colocalized regions. Now the question is this by random chance that this overlapped? Because it doesn't seem like they've got the same structure. Or is this actually structural? So there is something in the inherent molecular structure which led this to produce some overlap. Now the way that, again, Costas came up with I think Manders, oh no, it was Costas. Yeah, Costas came up with this idea where you take your one channel, you keep, let's say, the first channel fixed. You don't change one of the channels. And then your other channel, you just randomly shuffle the pixels. You just scramble this image around. And then you scramble them, calculate what is the Manders correlation coefficient now, scramble them again, now Manders, scramble again, now Manders, and you do this how many ever times. Let's say we do this 100 times, we scramble and then calculate. And then we say that if we ask the question, how many of these randomized pairs produced a correlation that is less than the one where it wasn't randomized. So in other words, if it was random, what would we expect the correlation to be? And if it's the original, which is not random, what is the correlation then? And if 95% of the ones that I scrambled has a worse correlation than the structured one, the original one, then it means that there is actually structure that led to the correlation and not just randomness. So hopefully that makes somewhat sense. But I compare the correlation between random scrambling and not scrambling, and if the scrambled one is worse than the non-scrambled one, then it means that the structure contributed to the correlation. Okay. So just to demonstrate this in Fiji, I can open up my ELA cells, and you don't need to follow along here. Like I said, you have a macro that does all of this for you. There's the ELA cells. Okay, so I first want to split my channels. And something I haven't been saying is, whenever you make changes to intensities here, it's going to have a much more substantial impact on the colocalization, because what colocalization does is it compares intensities. So I can't do some non-linear operations to my intensities. I can't boost the one intensity in a non-linear way compared to the other one. I can't do gamma correction, for example, because that's a non-linear operation. And then I will make the correlation either appear better or worse than it is in reality. So I can apply some filtering, I can remove some background noise, and all of these things are fine, but I don't really want to change intensities too much here, because that's going to skew my metrics. Okay, first thing I'm doing is I'm just splitting the channels. I don't really want a nucleus. I want to know how these two structures overlap with each other. And I think if I press I can see that, okay, no, it's not there. But anyways, I first want to clean up these images. Oh, come on, a little bit. So I will just do that background subtraction that I was showing earlier. So I think I chose like 10 or something. Let's just call that sufficient, because I don't want the background to add to my, oh, this correlation there when it's clearly just background noise. So I want to just look at the structures. That's the thing that I want to ultimately compare. And this one, background subtract, yeah, let's just also leave it at 10. You can always play around with this to get it more optimal. And now I've got two somewhat clean channels that I can now determine my colocalization on. So plugins, where is it? Let's analyze. Colocalization, coloc2. You choose the two images that you want to analyze. You say you want to do costas thresholding, the threshold regression, I mean. And then some other things that I will mention some of them just now. Click OK, it starts running the colocalization in the background. And again, you can do this with Z-stacks. The larger the images, the longer it takes. It can take sometimes quite long to actually calculate this. Come on, hopefully it will actually pop up. There it is, okay. So there's my scatter plot. I can't unfortunately make it bigger, but there's not a very strong correlation between it. I must interpret it in the context of the images that I have. These red dots are clearly, I think they might be autophagosomes, I'm not really sure, or it might be lysosomes, I'm not sure. But the point is that they are not strictly speaking associated to the mitochondria. They are just kind of floating around. Some of them are, but it's not very strong, and therefore this makes sense for what I'm actually observing. So I can now then look at the metrics here, and I can see, for example, Pearson's correlation. Sorry, let me just try to make this a bit bigger. Oops. There you go. Pearson's correlation coefficient, there it is. It tells me what it is above the threshold. In this case, it is actually negatively correlated, but only slightly. And then if there is no threshold, there is no, also almost no correlation. So they are not very strongly associated to each other. Let's look at the percentages for Manders. I would typically not really look at above 0 because there's too much structures or background noise that's considered then. So usually you want to look above the thresholded value for Manders. And yeah, I can see it's about 50% are overlapping. So yeah, somewhat associated, but not very strongly. Now, clearly you can see that this is, I'm getting a value, not very clear how to interpret this. What I can do is I can have lots of different image runs, different treatment groups, and compare these values. But yeah, they're not giving me a full picture. So we're still going to look at how do I get a fuller picture. But let's just, for now, go back to the slides. Okay, just before I get to more of that, just need to comment on these scatter plots. If you've got two structures that are very nicely, perfectly overlapping, then you can see that there is they obviously have a nice straight line on the scatter plot, and it would have a very high correlation coefficient, which is obviously great. But what if the one intensity looks something like this? So they are still, in my opinion, perfectly correlated. It's just that the one intensity wasn't as uniformly increasing than the other one. So now the scatter plot looks like this, and Pearson's now suddenly says, no, it's a pretty low correlation, or relatively low correlation, where they're still perfectly aligned. Like they're still perfectly associating with each other. It's just now it looks less. So how do we fix this? Now, there's a clever way where you say, instead of just trying to model it like this, instead of looking at intensities, let's look at the rank of the intensity. So we simply rank them. We give them a new rank number to say these intensities happened in this order. So we give them the order name, and then if we've got two intensities with the same value, we just split the difference, like 3.5, 3.5, before we go to the next one. And if we do this, we then, this is maybe slightly dim, we can turn a curve that looks like that into something that looks basically straight, because now we care about the rank and not about the actual intensity. And there we've now straightened our correlation curve, and the Spearman's coefficient then gives us again a very high correlation. So Spearman's is usually a more useful way of looking at well, it's at least a good one to consider alongside Pearson's correlation coefficient to see if the values are maybe slightly skewed. If it was to be aligned, will they show strong correlation? In other words, is one structure overlapping with another structure, a little bit less caring about the actual intensity values. Now what happens if I've got something like this, where the intensity goes up and then it goes down again? Here Spearman's breaks. Spearman's doesn't actually give a good result. This is the just this normal scatter plot. This is not the Spearman's scatter plot. But the reason why this doesn't work is if I was to align intensities, I would then rank the intensities in the incorrect way. The one increases linearly, the other one increases and then decreases. So if I rank them, the center part would be ranked before this rim, and then they will say, okay, well, they're not really aligned. So yeah, Spearman doesn't work in a weird intensity case like this. So just in summary of what I've been saying, if you know that things are linearly correlated, you can see it in the scatter plot, then you know how to interpret Pearsons. If they are increasing, but they're like monotonically increasing, but they're not necessarily in a straight line, you can fix for that with Spearman's. And then if the two images are not correlated, you should be able to see it in the scatter plot, and then the metrics should confirm that. So it's a multi-layered analysis colocalization. You look at the image, you look at what is actually seen as colocalized in your perception, you look at the scatter plot, what does the distribution actually looks like, how do I interpret the metrics in the context of the scatter plot, and then in the context of my image. But we can still do better, and I'll hopefully get, here some of the work that we've done here at Stellenbosch. Basically all of this that I'm going to share now was part of my PhD work, and then we're going to take a quick break after that. So we have all of our data captured perfectly in 3D, but historically the analysis wasn't always done in 3D. It was kind of at some point in 2017 at least when we did this work, it was not uncommon to say, okay, we've captured our images, it's a little bit hard to do the colocalization analysis, so let's just flatten them, do a maximum intensity projection, and then we compare the colocalization. There are a couple of problems with this. The one problem is that you are then introducing intensities onto a certain position where it wasn't necessarily at a position. So if you, let's say for example, I have a structure that goes like this, and then there was some green intensities at the top, and then nothing at the bottom. But now in the other image I had some red intensities at the bottom and no red intensities at the top. As soon as you flatten this, it seems like where there is green, there is red, but in reality there never was. So the Z does contain quite a bit of information that you really shouldn't throw away, especially for colocalization analysis. Another problem is if you look at an MIP, if you make a region of interest selection to analyze how does it look in a region, you're basically taking like a cylindrical cut or whatever, just straight through your entire structure. But the structures in a cell isn't nice straight lines, they are three-dimensional shapes. So you actually want to make selections a little bit more precisely. So we developed this system, it's kind of a two-stage video. The one was just to take our Z stacks and then visualize it, and it was a VR-based system because we understood, first of all, that the cell has got some complex structures to it sometimes, and it's hard to fully appreciate it when you just see it on a screen like this. You actually want to have the intuition of, I see my cell in front of me, and know what I'm actually looking at. So we bought this interaction things. The hand tracking, we did tests. The hand tracking was apparently, well, according to our tests, more productive than using a gamepad controller. But in my experience, it led to quite severe arm fatigue over time. So yeah, not necessarily a way that, even though it feels futuristic, not a way you actually want to analyze your samples. So you can see, so you can see I make a selection. This is a very crude selection, but I can just lean around it and then see, did I make the correct three-dimensional selection? Or instead of trying to draw it with a mouse, just draw it out, paint it out with your finger. Okay, so yeah, you can now just see the scatter plots and the metrics that were calculated. So yeah, this is the second version of the system. I still had some hair back then. The quality is a little bit better here, and you've actually seen the sample. I've used it as one of our examples. So you can, this is a general thing, you can change the opacity, the thresholds and stuff just for visualization purposes. And then you can visualize the colocalization in various ways that I've just showed you earlier. And for example, in this case now I can easily see there is these hot spots around the perinuclear region. So I want to analyze that. What is the colocalization actually going on there? And now I can make these three-dimensional selections, which you have these ellipsoids which you can stretch and squash in any dimension. And then I can select the outside, put another one inside to cut that out, and then I just select the rim of the cell. This is not necessarily a perfect selection, but it's a more accurate selection than if I was to just take a cylindrical cut through, because then I would throw the top and the bottom away of my nucleus, which I don't necessarily want to do. You could also first crop out a section, then make a more precise selection, some turbulent strands here that we wanted to analyze. Just want to say the Z dimension here is a little bit exaggerated. There's a slider there that you can tune it. I don't know, for some reason for this video I stretched it out. You forget why you do things and then later you regret them. Okay, so analyze the colocalization. This is what it looks like. You can see the thresholds determined, etc. Okay, so we did that. We bought this, we used this then for analysis, and then we compared, if we take our Z stack and we do an MIP, or we take this, keep the data three-dimensional, how does the analysis compare? And made several selections, made these cylindrical selections to show how that compares to these ellipsoidal selections. Just if there is a difference, does it matter? Membrane selections, either cropping out the membrane perfectly or just taking some samples in different vicinities of the cell. Yeah, it's just all the data, some protein aggregates, turbulent strands. Here's actually the results that matters. So this is the analysis that were done in the different regions. In 3D on the left, and in 2D using a maximum intensity projection on the right. And we looked at 3 different time points of control, 6 hours, 24 hours, so it's the same. So the time points are the same in both cases, it's just 3D and 2D. Now if you were to look what happens across time, you will see that in the 2D case, it showed for the different regions, it showed, generally speaking, let's just look at the microtubule. This is the one which confused my co-researcher. He was saying that he's not expecting it to change at all over time, but he's seeing this increase, this statistically significant increase in the colocalization in the microtubule. And then after we do it in 3D, you can see actually there is no change. It stays basically the same. And this is just, really just doing it in 2D versus 3D. Yes, that's your Gauss one. Some other things to notice, here is an average increase. Usually it goes up, the values, with time. In the 3D, it actually generally goes down. So very opposite result. In 2D, there is no statistical significance that we could find between these different time points. In 3D, taking all of the data into consideration, most of them were statistically significantly different from the control. So hopefully the bottom line is, if you have 3D data, analyze things in 3D. If you do things in 2D, what does the stats even mean? Because as I was saying, you might be conflating things which shouldn't be put together. Now, I've been complaining a lot throughout this entire time about the visualization is limited. We don't like to just look at the yellow and make some interpretation from that. We don't like to show it in this harsh white because that's overemphasizing certain things maybe. So this is another method that I worked on where we give a new visualization, a new qualitative visual representation of the colocalization, where bright spots in this case is trying to emphasize the regions of high correlation, and the dimmer areas is where there's lower correlation. So for example, when two structures associate, if they associate strongly, the intensities are the same, relatively speaking, correlated. I want to show that is the point of interaction. And then where they are slightly less strongly interacted, I don't want to highlight that because that's maybe some resolving power issues or some non-specific probe or something. So I want to emphasize this correlation. I'll show you some more examples of this. I'm not going to go through the technicalities of how this method works, but some ideas of what it's trying to achieve is it first finds this regression line of all of your data on the scatter plot. What is the regression line? There's a very specific way in which this needs to be done. It's not just linear regression. But once you find this regression line, then you can say, okay, well, let's just assign perpendicularly different color map values to this. If I land somewhere on this line, if the voxel is in the scatter plot somewhere on this line, I'm going to just assign this color intensity to it. So just lower intensities are dimmer, higher intensities are brighter. But now I want to emphasize the correlation. So how I do that is if it's far away from this regression line, I'm going to attenuate it. I'm going to give it a dimmer intensity, where if it's very strongly correlated, then I want to give it a brighter intensity. And then the last step is if something is very clearly not correlated, this could be coincidental, I want to remove that as far as possible. So I'm putting some general thresholds in to say some distance away is the only thing I will consider colocalized. And if you do that, then this is some of the results. You can see, first of all, that dot there is the mean, the joint mean for the two channels. And there's the regression line. This is the extent that I cut out this, I removed everything above that line, removed everything above that maximum so that I can get the best spread for my color map, and I get these types of visualizations. Yeah, maybe just to go a little bit more into this one. So the point here is that, autophagosomes are green and the magenta or purple is tubulin, and the autophagosomes are supposed to move along the tubulin. So you would expect there to be some interaction, but very, very slight. And you can now see if I was to visualize it in this way, there's probably interaction, but I can't see it visually. Here it's like, okay, I've got these hotspots, but I don't know how correlated it is. And then now I'm just visualizing it, oh, these two are strongly correlated. So I need to interpret this together. With the intensity with my qualitative field, these two areas is where I would have strong interaction, and then everywhere else it's a little bit less. And you can obviously look at a more macroscopic view of this as well. Just another application of this is with looking at mitophagy, basically, where we've got our autophagosomes, lysosomes, mitochondria, and if we just calculate a single PCC value for this, we get a pretty low value because there is very little interaction. It's only in the perinuclear region. But if we use now this visualization method of mine, you can suddenly see, okay, well, first of all, I get an intuition for where are the bright spots, where's the areas of high correlation, but it's more interpretable than whatever this mess is that's going on there. Also, if there is not a lot of interaction, you can see PCC is quite low. You can see, relatively speaking, that the correlation values assigned to it is also relatively low. It still shows you where they are, but they're not as bright spots. Then if the correlation value is higher, then again, it shows more bright regions. And yeah, it's just a helpful additional tool to analyze localization. It is available. It's currently implemented as a Python library. I'm still hoping one day, once I get time, I will put it into Fiji as well, but for now it's just Python. It was implemented in 2018, 19, somewhere there, so it's Python has moved quite a bit since then, so if you use the newest version of Python, use news libraries, it probably won't work. So you need to use old software, then it will run. If you really want to use it, speak to me. It's probably easier than to try to get it figured out by yourself. Okay, so that's the talk on colocalization. Any questions or comments on that?